Hi, I'm Ted Haptegaber, founder and producer of Live Talks Los Angeles. Thanks for joining us. Since we started over a decade ago, we have brought you hundreds of conversations with storytellers, writers, actors, musicians, humorists, chefs, and thought leaders in business and science. You can watch and hear most of these in our YouTube channel and our podcast. For details, visit livetalksla.org. And now, here's the show you've tuned in to see.
Well, Sid, welcome to LA.
Well, thank you very much.
And congratulations on the release of the book. I told you before I was flying the past 2 weekends, and it's everywhere in every airport bookstore.
Oh, well,
So,
good for airports.
the book is The Song of the Cell, and I want to start with what a cell is.
A cell is the least or the smallest autonomous unit of life. You and I are made of cells. Everything that's living is made of cells. It is the single binding element that runs through life. Maybe I'll say a little bit more. So, you know, physicists always make fun of biologists. It's an old story. And biologists eventually get tired of it. But what's interesting about biology is that there are three themes that are sort of grand unifying themes in biology. The first is evolution, evolution by natural selection. The second is the universality of the genetic code that we share with bacteria and elephants. And the third is cell theory. So most of these discoveries were made by the 1950s. So we're pretty happy. We've found our grand unified theory, and the physicists seem to be still searching for it. So seems as if one of the two camps has won the game.
Take us into the deep past that you start the book with. And let's go to a time before we knew cells existed. What were the prevailing theories of life at that juncture, and what were some of these signal moments in the discovery of the cell?
Well, the deep past, I mean, you know, the question of what flesh is and what flesh is made of was of course an ancient question. People like Aristotle and others battled with that question and thought a lot about it. And they thought it was continuous. They thought that, you know, we were slabs of meat. And then this was the theory for the longest time, and they would look at organs, and organs themselves were also considered slabs of meat sitting inside slabs of meat. So there was the idea that it was all continuous. It was just one big continuum, and the self, the body, was just a big continuum of flesh, kreas, as the Greeks might have put it. And then all of a sudden comes this guy, Robert Hooke, first. Now Hooke's a strange character. He's a great. Hooke's an incredible polymath. He is an inventor. He's an architect. He's a scientist. He works on mechanics. He works on optics. He makes telescopes. He makes microscopes. He's just all, you know, his intellect is just roving. What's interesting about Hooke is that I use this phrase in the book, his intelligence is phosphorescent and elastic, like a rubber band that glows as you stretch it. and there's a little bit of a pun here because, of course, Hooke invented the laws of elasticity. The laws are named after him. But anyway, Hooke finds a microscope or creates a microscope, and he cuts tissue with it. And in a piece of cork, he cuts a piece of cork, and he finds, to his amazement, that cork is not just, again, a slab of tree, but in fact is made of what he calls a great many little boxes. And he draws them, and he finds them, and he draws them, but it's not clear he pays a lot of attention to it. He's looking basically at cell walls, and that's how he's finding them. And then a little bit later comes this other guy named Leeuwenhoek, who's a Dutch trader, not a scientist, not trained in the language of science, doesn't have any formal training in science, and he wants a microscope to look at the quality of cloth, and because he's a cloth merchant. So he invents a different kind of microscope. He makes about 500 of them. He's very different from Hooke. As I said, he's not trained as a scientist. He's secretive. He's very reclusive. And he makes this microscope and looks at a drop of water. And in that drop of water, he finds these floating animalcules all over, all kinds of things, probably protozoa, protists, other organisms, etc. And he's totally shocked because people have been looking through the telescope and found massive universes. And Leeuwenhoek all of a sudden begins to find these amazing creatures floating in water. And so he begins to write about them, and he writes to the Royal Society. And ironically, Hooke is the president of the Royal Society. So Leeuwenhoek and Hooke realize that they've sort of stumbled on the same idea, that living creatures that they're living creatures that are autonomous, in Leeuwenhoek's case, single-celled, single boxes, and in Hooke's case, in a piece of cork made out of thousands of little boxes.
And you have to imagine, this is shocking, right, at that moment in time, to look through this microscope and see all these tiny animals.
Right. So I tried to build one in the middle of COVID. I tried to build one of these Leeuwenhoek single scopes, and I'm a terrible builder, but I managed it eventually. And it's when you make one and you see one, and you see you put a droplet of water on one end of it, it's kind of astonishing. You have to hold it up to the sunlight because there's really no source of light in there. It doesn't look like a microscope. It looks like something that you would find in an antique shop. But it's amazing. You can see this whole tiny world in that microscope. And
so these guys see, in one case, the cork cells, and in another case, these tiny microbes. And then, as you describe it, significant time goes by before others know what to do with these observations and realize that these are the basis of all life. The origin of cell theory really is. Tell us about how that transition occurred.
So there's sort of a gap in the middle. So now we're moving from the late 1600s, early 1700s, when Hooke and Leeuwenhoek are still looking down microscopes. But and they're seeing the same things over and over again. They're seeing, these moving life forms, protists and other life forms. And then there's a sort of gap in the middle when microscopists, and I name a lot of them, people who aren't sort of the big canon of science, they start looking at tissues. So not just at single protists or not just at single-celled animals, but at real human tissues. And they begin to find, once again, that they're cells. But they don't make that final leap, which is the first tenet of cell theory, And this first tenet is that all organisms, plants, animals, every organism is a cell or made up of cells. And that begins the book. So there's an evening, I'll take you back sort of, we only know this from one letter that survives. But there's an evening in Berlin when two characters, Schleiden and Schwann, a zoologist and a botanist. So they themselves are interesting characters. One of them has tried to shoot himself. And he has a big scar. He misses, thankfully. So he has a big scar across his face. And they're sitting, and they're lab mates. They work under Johannes Müller. So they're sitting there thinking about sort of, how life is organized. And essentially, through a convoluted series of things, they both realize that one of them is studying plants, one of them is studying animals. They both realize that in both animals and plants, the formation of the organism is a cellular formation. They're formed through cells. And so they make this very bold claim that, number one, the cell is the smallest unit, autonomous living unit, and number two, that all living creatures are made out of cells. Again, very bold claims for their times, and that lays the basis for contemporary or modern cell theory.
And it invites the question of, where do these things come from?
Right.
Where do cells come from?
So now, Schleiden and Schwann are students of Müller's, and Müller and a lot of other folks are sort of transitional characters. And by transitional, they are scientists in the modern sense, but they also inherit a lot of beliefs from the past. And one of the beliefs is that cells crystallize out of vital fluids. And if you think about it, I mean, it's not sort of a crazy, absurd theory. And because they see the birth of cells, they see the appearance of cells. And the only other thing that they've seen in the past to some extent is when you take a chemical, you have a fluid, and all of a sudden crystals form spontaneously out of the fluid. So you could imagine, it's not so illogical to think that out of vital fluid, these cells would suddenly sprout and come across. And that leads us up to, the 1800s, when Rudolf Virchow, the pathologist, borrowing from previous ideas in pathology, makes the third claim. And the third claim is that all cells come from other cells, omnis cellula et cellula. Now, that's an incredibly bold claim as well, because it says there's no vital fluid, there's no crystal, all of this stuff. Cells come from cells. That means that you and I had to come from the single cell, the zygote. So that's an incredibly important idea. I'll just extend that idea and then tell you about Virchow's second and third postulates, which will bring us up to speed in terms of the fullness of cell theory. So think about this idea for a second. There are two things about that idea that are very important. Number one is that you and I come from single cells. That means that the instructions to build you and to build me must be in that single cell. In fact, ironically, it's the sperm is the smallest cell and the ovum is the largest cell in the body. And there's a reason for that, right? The reason is that the sperm is just a carrier of DNA. It has no other function. It's basically DNA with a big tail attached to it. And its job is to get to the egg as fast as possible. The egg, on the other hand, has to carry a lot of information, epigenetic information. So not just genetic information, information that is beyond the genome and above the genome. So it has to have positional information. It has to know up from down, otherwise you would be upside down. It has to know left from right, otherwise your heart would be on the wrong side of your body. It has to know, right northeast from northwest, otherwise, your body would be misassembled. So all that information is partially, not complete, but partially located in the egg. So this idea of an omniscellular et cellular leads to the whole idea of human and animal development. How do you develop from that single cell into an organism? And that's the basis of developmental biology. And even today, we're finding out how that happens.
One of the other things the egg has
Yeah.
is the mitochondria.
Yeah.
And that's one of the organelles, the so-called organs of the cell, in a sense, that you walk us through in a series of chapters.
Yeah.
Tell us about your favorite organelles and
Right.
why you love them so much.
No one's ever asked me that before. Let me see, what's my favorite organelle? I mean, people generally say, what's your favorite musician? Or who's your favorite musician? But my favorite organelle, well, mitochondria have a big, are a little bit of an easy answer because they are obviously so vital to to everything that happens in the cell. And as you know, the theory of the origin of mitochondria is very interesting, and that's why I love them so much. So Lynn Margulis, who happened to be married at some point of time to Carl Sagan, so power couple. Lynn Margulis was among the first to propose that mitochondria arose as a consequence of one cell swallowing another cell. So the mitochondria themselves at some point of time were cells that were suddenly acquired, not suddenly, but through evolution, acquired the capacity to generate energy from metabolic sources such as glucose. And generate energy in a way that was very particular, generate energy using oxygen and glucose. That was called the endosymbiotic hypothesis. Endosymbiotic meaning that, again, the larger cell swallowed the mitochondria, and essentially the mitochondria became symbiotic with the larger cell. And she was considered a completely crazy character, ridiculed, until now, not now, but more recently, we've figured out that this hypothesis, endosymbiotic hypothesis, is absolutely correct. Mitochondria have their own genomes, which are quite different. They resemble bacterial genomes. And it's almost certainly true that that's how they came about, through this ancient swallowing, probably about 2 billion odd years ago, this ancient swallowing of one cell by another cell.
And that may be significantly into the history of life on Earth, that event occurred. If we go way back to the first cell.
Right.
You talk in the book a little bit about the origin of life in the first instance, and then later on you also talk about the origin of multicellularity.
Right.
And I wonder if you could walk us through both of those mysteries and some of the experimental work that people have done to try and poke at those.
Right. So, let's go through the thought experiment, which is very interesting, actually. So, if you were to take every single living creature on the Earth, so you, me, the hundreds of millions, potentially billions of bacteria that live within us, the plant that you saw outside, the lizard that you saw before that on the plant, the bird that you heard singing this morning, the snail in your garden, all of us, if you rewind the biological clock, or the real clock, time clock, 100 years, 500 years, 1,000 years, up to 3.5 to 4 billion years ago, came from one cell. So we all came from that one single cell. And if that cell had not divided, life on Earth would not exist. That's called the last universal common ancestor. We call her Luca. She decided to divide and give rise to all these cells. Now, what do we know about this first cell? We know very little because, of course, you can't really rewind the clock. You can only do experiments. We know that almost certainly that cell, its information was not carried in DNA but in RNA, a molecule that's related to DNA, but it's a single strand, not a double strand, not a double helix. And it had to have also a duplicator. Now, interestingly, RNA can itself be a self-duplicating molecule because it has, I said we don't have self-duplicating RNAs yet, but has properties that can make activities happen. It's not just an inert sort of strand it's just not an inert molecule, it's not an inert strand. So either itself or along with the duplicator, it had to be present together. But if they were just those two things, then they would potentially drift apart and not be able to create a metabolic milieu, something that was contained. So there had to be a membrane. And that membrane, we know, was likely made out of fats. So a fatty membrane, RNA inside it, and potentially a duplicator, whether itself or otherwise. And what's interesting is that you can make these things, these protocells in the lab. So Jack Szostak and others have shown that you can make these membranes in the lab with fat, with particular kinds of fat. It's not just any fat, but a particular kind of fat called a phospholipid. And if you keep adding more of these phospholipids to it, you can actually make these. Now, this is not life, but this is just a bubble. You can make these bubbles grow. You can make an internal milieu within these bubbles. And if you keep adding more and more, they actually split and become two. Naturally, just by energetics, just by thermodynamics, they become two. And then 2 becomes 4, and so forth. So we think that somewhere in that process, we've captured the protocell, the first cell that ever existed. So that was the first part of your question. The second part of it should give you some chills up your spine to know that all of us, that the entire living kingdom came out of that one cell division. 3.5 billion years ago.
And just to be clear, we have not been able to replicate this. I mean, we've never made life from not life.
Correct. We've never made life from not life. Absolutely correct. We've only made, the closest we've made is made artificial genetic material, put it into a living organism, and made life, but we've made life from life.
All cells come from cells.
All cells come from cells, exactly.
Except one cell.
So there we have it. Now the second question is also very interesting, because you asked, why is it that many-celled organisms exist? So first of all, you have to realize that single-celled organisms, bacteria, protists, protozoa, et cetera, are extraordinarily successful. They live in places that you and I could barely imagine living in. Thermal vents, which are really nearly boiling temperature, freezing cold tundra, toxic environments where you and I would die in seconds. They colonize them and live perfectly well in them. So single-celled animals, single-celled bacteria, and other animals are very, very successful, phenomenally successful. So the question then arises: well, why is life not single-celled? Why is all of life not single-celled? And for a long time, biologists thought that it was a big transition. The transition from single cells to multicellularity was a big transition. But it turns out that if you look genetically and with other pieces of evidence, it turns out that there's a force that's driving single cells to become cooperative units of multiple cells. And it happened not once, but multiple times independently in evolution. There's evidence to suggest that it happened several times in evolution. In other words, this drive was present in several instances. You can replicate some of that in the laboratory. The book goes through in one experiment where a researcher replicated this in the laboratory. And we don't exactly know why. Why is it that these single cells, which were incredibly successful, want to congregate and become multiple cells? And there are many ideas about it. One idea is that it's to escape predation. It's to escape being eaten up. It's harder to eat a big glob of cells rather than a single cell. Another idea is specialization. So you can have one part of the cell be a digestive unit, another part of the cell be a unit for movement, yet another part of the cell be the metabolic center of the cell, et cetera, such that you can have cellular specialization. Yet another theory is that it gives directionality to the cell such that it can move towards food and move away from predators, and so forth. But nonetheless, the drive to become multicellular was very strong and repeated over and over in evolution. And that too should be a matter of great interest because you can wonder why. And also, I think there's a moral in there about citizenship and citizenry, that somehow or the other, evolution likes, I don't want to put an anthropomorphic touch on evolution, but the drive in evolution and natural selection is towards citizenship. It's important somehow.
Well, let's zoom way forward in the history of evolution to us.
Okay.
And the sort of citizenship that you talk about in the book that's going on among our various cell types is so extraordinary. I think of my first exposure to it as I started investing in biotech and first learned about adaptive immunity. You go through the way that B cells differentiate in response to invaders. Mm-hmm. Tell us a little bit about that. Give us a snapshot of how B cells work and then maybe how T cells work. A minute on each. It's a tall order.
So B cells are fascinating. B cells are basically, their job is to produce antibodies. Antibodies are missiles, molecular missiles that attach themselves to bacteria or germs or viruses and neutralize them or capture them so that they can be cleared out by cells, other cells in the body, and removed. So they're missile-producing cells. And the way they make their missiles is interesting. They make their missiles by basically shuffling genes around until they have unique combinations. So every single B cell is carrying a unique missile. And when it catches, when that B cell finds a cognate bacterium, it starts differentiating and becoming a professional missile-secreting cell. A T cell is very different. T cells is what I study quite a lot. A T cell is not a missile maker. There are two or three kinds of T cells, but one kind of T cell, the killer T cell, goes and sniffs every cell in the body and asks the question, is there a virus inside you? Or ask the question, do you belong to this body or are you from somewhere else? And it has specialized molecules to ask that question. And it asks the question, it goes around asking that question over and over and over again. And if it finds that there's a virus inside you, it has a mechanism by which it'll kill that cell. If it finds that it's not part of your own body, again, it has a mechanism to kill that cell. That's one kind of T cell, so-called CD8 or killer cell. The other kind of T cell is called a helper cell, although, as I said, calling it a helper cell is calling Thomas Cromwell a bureaucrat, because it's an incredible cell. So that's called a CD4 cell. And anyone who's knows about sort of popular science and popular literature knows that CD4 cells is what HIV infects. And if the CD4 cell collapses, or the CD4 cell count collapses, then you get AIDS. And so it's incredibly important because the CD4 cell, the CD4 T cell, doesn't go around sniffing. It goes around coordinating the entire immune response, making B cells talk to T cells, making B and T cells talk to other immune cells, and therefore coordinating, sort of machinating the entire immune response.
And so that is one example of this extraordinary complexity in just one or two cell types is the same story across all of the cell types. And you go through a few examples of the cells that make up specific organs, nephrons, hepatocytes, and so forth. And each of them is a compelling tale in its own right. But where you begin the book is with this description of Emily Whitehead. Mm-hmm. That's one of the first vignettes. And it speaks to now this new actor in this incredibly complex ecology, which is us and our technology. And it appears that we are at the verge of a major transition in history where we're not merely the output of this ecology, but are starting to play a role in how it works. And so could you tell us about what Emily Whitehead benefited from and how technologies like it are now reshaping what it means to be human?
So the book has a provocative subtitle. If there is the subtitle, part of the subtitle is The New Human. And so the question is, who is the new human? And I make it very clear that, we've gotten used to, partly because of popular sci-fi, the new human being sort of like an infrared-equipped, prosthetically enhanced creature. What I call Keanu Reeves in a black muumuu, sort of a Matrix character. But in fact, walking amongst us in this auditorium, there are people who've had a bone marrow transplant. Their blood system is not theirs. They're actually a chimera. Their blood system comes from someone else. There are people who will soon be walking amongst us, maybe already walking amongst us, who have electrodes inserted to stimulate certain cells in their brain so that they can get relief from diseases such as Parkinson's or maybe recalcitrant depression. There are people in whom we are transplanting genetically engineered cells, and that brings us to Emily Whitehead. So Emily Whitehead had very lethal form of cancer that relapsed, called acute lymphoblastic leukemia. Her chances of survival without any therapy was 0%, so she was basically given up for dead. And she got a transplant. She was transplanted. T cells were extracted from her body, genetically engineered to attack the cancer. And remember, T cells usually go sniffing around to ask the question, are you foreign or are you not foreign? So basically, this T cell was engineered to recognize the cancer as foreign and then regrown in a laboratory. Half of it was stored in a big vat, which happens to be called Krusty the Clown. And half of it was returned to her body. And those cells became expanded, became highly active, almost killed her in their mad glee to kill cancer cells. But they killed the cancer, and Emily is still alive. So this was when she was 7. She is now 17 and applying for college and has been cancer-free. So really an incredible miracle of science. And we did a lovely segment on CBS last week with her. And I've been friends with the family for years, and I would encourage people to watch it. It's one of these stories that really restores some faith in what science can achieve and can do if it's applied in the right direction.
I think that friendship you mentioned with her and her family is emblematic of a broader observation that I would have from the book, which is that you love the science, but you love the people. You love the patients, you love the other researchers with whom you've collaborated or marveled at the discoveries of in history. And I want to know what it is that captivates you about those people on both sides of the equation, the inventors and the beneficiaries.
I just like telling stories. I think telling stories is very important. I think telling the stories of people is very important, taking them to the right place. I often go to places to just visit them, to be there. I went to, for the gene, I went to Mendel's Monastery. And I'll tell you a funny story about that in a second. But I like this. I like their stories. In a sense, this book is sort of an extended love letter to them. There was a lovely review today, which I couldn't read because it was firewalled. But it started with saying that this book is a love letter to a cell, which I thought it's a lovely idea. But it's also a love letter to all these people, to all the people, the inventors, the patients, the teachers, the nurses, the doctors, everyone. So I think it comes from a love of storytelling. Now going back to Mendel, just to finish up that little story. So I went to Mendel's monastery. These days is not particularly easy to get to. You have to. It's in a town called Brno, which is in the middle of the Czech Republic. It used to be a big town, but is now becoming a kind of a dwindling Central European town. So I took a train landed in Vienna, took a train from Vienna, landed up in Mendel's monastery. It's very hard to communicate, blah, 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 blah. And landed up, and there was a woman, and she said, but it's closed. And I said, but you don't understand. I've come from, thousands of miles to come and just look at Mendel's monastery. And she said, it's closed and there's nothing to see. But I said, that's okay. I just want to be inside. And she said, okay, well, you have to apply to get in triplicate. And I said, okay, I'm from India. I know how to play this game. I said, I stood before her. I said, I hereby apply in triplicate. And she was like, okay, now you've beaten me at your game. And so she let me in. So that was my trip to Mendel's monastery.
It's clear that you like heretics. And that's a common thread through these inventors, even people like Leeuwenhoek, who didn't know he was an inventor necessarily, or maybe thought he was the greatest inventor. But they all rebelled against some incumbent wisdom that proliferated at their moment. And I'm curious what you think the heretics of today are pushing against. We're almost certainly wrong about some significant orthodoxies in medicine or cellular biology. What do you think might be candidates?
So I'm careful about heretics. And I want to make a very important distinction here, which we can talk about. It'll take us the entire evening to talk about it. So I'll plant the idea in your head. I make a strong, bright line distinction between disease and desire. Disease is linked to suffering, and it is about suffering, and we recognize it as something that's linked to suffering. Desire is linked to enhancement or augmentation, and the desire to become something else, taller, bigger, I don't know, having some other qualities that we don't have. They're different things, and the lines between them are generally bright lines. because we recognize suffering. But I think that those lines are changing. So I think there are heretics who move in the direction of desire, and I think they're problematic heretics. So I describe, of course, in a chapter called The Tampered Cell, I describe the Chinese scientist who started making genetically engineered babies without informed consent, without telling the scientific community, and really created a ruckus in the world of science. And then there are people who treat disease and are also heretics, or people who make inventions that eventually will be used to create disease or to cure diseases, who are also heretics. So I make a strong distinction between people who fall on heretics who fall on one side and heretics who fall on the other. Now, your question is about about heresies in cell biology that we yet don't recognize. And of course, if we don't yet recognize them, how would I know? But but I can make some guesses. I think and the reason that this book is called The Song of the Cell is that there's a movement at the end of the book. There's a story which I tell, which is of a professor of botany who goes to the rainforest, and he takes a local kid into the rainforest with him, and the professor of botany is amazed because this kid can name every plant, and he goes after every plant and names every plant. And the professor of botany says, "Well, that's amazing. You can name every one of these hundreds of plants that are around." But the young kid is dejected. He said, I know the plants, and this is from Amitav Ghosh's book, but I don't know the songs between them. And he, interestingly, means it quite literally. He really doesn't. What he's saying is he doesn't understand the interactions that occur between the plants and perhaps the birds that sit on the plants and the insects that feed on the plants, etc., the entire complex ecosystem. So I think the new heretics of today are going to solve the problem of how these cellular systems don't just sit in isolation, but in fact talk to each other. And we've already started seeing some of this. We've learned now that this gut-brain axis, for instance, is becoming very important. Somehow or the other, our anticipation, for instance, of sugar already triggers a series of activities in our brain, which then feed back into our intestines. So I think those kind of long-range or short-range interactions between cells is one of the things that I think is going to really empower our understanding of not just the song of the cell, but the songs of many cells.
Does this connect to what I would call a nerd bomb that you drop in the book when you say sequencing is seduction? What does that mean? What are you fighting against there?
So it's a little bit of a nerd bomb, except for the fact that there's a long passage that precedes it. So there was this idea that after we started sequencing the human genome and started sequencing cancer genomes, that all of a sudden all these cures for cancer would just fall out of that process. That basically that we would find the answers to, we would sequence a cancer, find a matched medicine for that, and that would be the way that we would move forward. And that has been an incredibly sobering experience. That is, aside from a few examples, that has not worked out to be the case. And that's been very humbling for people who were big advocates of this theory. And it was a very seductive theory. Cancer is a genetic disease. If you sequence the genomes of cancer, we would find the mutations. The mutations would lead us to drugs, then the drugs would cure the cancer, right? Sounds like a wonderful theory, but that was all seduction. We are in trials, in real human experience, That has not turned out to be the case. What has turned out to be the case is that understanding the cell biology of cancer, understanding the cancer as a cell not living in a vacuum, but living in a particular home, metastasizing to particular places, creating particular ways or metabolizing particular nutrients, and depending on particular circumstances, create vulnerabilities for that cancer. And it's only by understanding the cell biology of that cancer that we can unveil those vulnerabilities and use them to make new drugs like immune therapies, or in Emily Whitehead's case, genetically modified cellular therapies that will kill the cancer cell. So not nerd bomb.
Yeah, corrected. What I take that response to mean is that you think the secret to cancer, in a sense, is in that song.
Yes, I do think the secret to cancer is in that song. It is in the interactions that, it's in the cancer cell, but it's within the cancer cell, it's in the interactions of the many parts of the cancer cell that we don't fully understand. And outside the cancer cell, it is the interactions of the cancer cell with the immune system, with the blood system, with its local neighbors, and so many other parameters that we don't fully understand, that if we did understand, we would be able to direct and are directing new therapies against this. For example, as I said, new immune therapies, new therapies to stop the cancer from pulling out blood vessels and feeding itself. You and I have been talking about, my interest in the metabolism of cancer, the fact that cancer metabolizes things differently, makes different, attaches different sugars to different proteins than normal cells and so forth. So inside the cancer cell and outside the cancer cell, the sort of malignant song of the cancer cell is something we need to understand before we start, going down this route of sequencing, which is very seductive.
Sid, before we open it up to questions, I don't know how we are on time, but Ted'll tell us. I want to understand the connection between everything we've just talked about, this story of cellular biology that you masterfully illustrate in the book, and the metaphysics that you hint at periodically. I mean, you bring Aristotle into this, you bring Hinduism into this periodically in the book. For you, as not just a scientist and a clinician, but as an artist in your own right, where is the soul in this work for you?
I mean, this is a very unusual book because this book is written as a series of almost, you can think of this, unlike Emperor or unlike The Gene, you can think of this book as a series of short stories and a series of love letters to individual cells. It's a slightly bizarre organization, but one I've sort of learned to love. This book was very hard to write because when you write a book, chronology is your friend. So you write a timeline, and you say, I'm going to start at the beginning. You could start at the end, actually, if you wanted to, but that would make you a different kind of writer. But you start at the beginning, end at the end, and that's how you write a book. But the problem is that cells are so unique and so different. And there is no, if you followed the chronology, you'd have to go into 1,000 different ways. People were exploring the brain while they were exploring the kidney. People were exploring the heart while they were exploring so you'd have to somehow figure out a way. So instead, each chapter becomes a short story, or as this reviewer put it, each chapter becomes a love letter to a particular cell. So there's a love letter to the pancreatic cell, and one to the T cell, and one to the B cell, and one to all these other cell types. So this book is organized as a series of stories. And the soul of this book, I think, is exactly as I said, is my decades-long love affair with cells.
Why don't we pause there and open it up?
Great. If you have a question, please raise your hand and I'll bring the microphone to you. Just a quick reminder, around here, questions typically begin with a W or an H. They are generally short. There is no such thing as a two-part question, and tonight, only D.A. Wallach gets to ask the follow-up questions. Who would like to go first?
If these cells can live anywhere, any harsh environment in the world, do you think there is a cell life on other planets in our solar system?
Well, I mean, numerically or theoretically, just given the number of exoplanets which resemble the circumstances of the Earth, just numerically, it would be unsurprising to me if there was not another cell type in another planet. Just by numbers, it would be a little bit surprising. Will we ever find one? Probably not, just because they're so far away. If they've somehow managed to send out signals, maybe we'll find a signal. But will we ever meet one? I don't think so, just because everything is so far away that it would take us time beyond time to get there. Now, there is a theory, which I don't subscribe to, but there's a theory that the origin of the cellular universe on Earth is actually from Mars, which cooled down before the Earth, and then became colder and colder and colder, but cooled down before the Earth, and some piece of it was knocked off and landed on the Earth with cellular life in it. But Mars then became colder and colder and colder and colder, such that it could itself no longer sustain cellular life, but Earth can. I don't know if I'm not the biggest believer in that theory. It's just kind of a just so story. It's very hard to prove or disprove. Certainly, we haven't found any evidence for any of this. But again, numerically speaking, I think it would be hard to imagine that there wasn't cellular life in other planets. What it looks like, I can't tell you. What it breathes, how it metabolizes, I can't tell you any of this. Is there a chance we'll ever meet it? Probably not.
I have a follow-up.
Yeah.
Not a joke. If, as you contend in the latest nerd bomb that you dropped, there are aliens.
Yes.
Just to be clear about the answer to that question, you said there are definitely aliens. Would you assume that aliens also have cancer?
Yeah, so that's another very good question. I think that the laws of evolution, some kind of organization, whether it's cellular or not, I can't imagine a non-cellular organization, but I suppose there could be other mechanisms of organizing tissues and structures. And I can't say anything about genetics because it's impossible to extrapolate about why or how the universality of the genetic code is really a consequence of evolution. So it really leaves us with evolution and cell theory. So the quick answer is cancer, and wrap your brains around this, but it is interesting. Cancer is the one disease which distorts and exploits all three of the fundamental pillars of life. So it's an evolutionary disease, so it evolves, and that's what makes it very hard to kill. It's a cell biological disease. It uses the cellular physiology, distorts it, and it is a genetic disease. It alters the genetic code. So in a bizarre way, it's the only disease that takes the three fundamental pieces of human, of all life, and twists them around and twirls them to its own advantage. And it's the only disease that I know of that takes all three of them and does so. So if that's the case, I would imagine that aliens also get cancer, along with dreaming of electric sheep.
Fair enough. Next question.
How do scientists explain why the therapy worked on Emily, but perhaps not another young lady?
Yeah, so we've been looking very hard at why it works for Emily and not for everyone. Generally speaking, it's pretty successful. So the success rates for CD19, which is what Emily was treated with, CD19 CAR T therapy, is now hovering around 70 to 80%. So these are very difficult cases, right? So remember, these are cases that have relapsed after usually 2 years of chemotherapy. There's a 80-90% cure rate for this with chemo alone. And so Emily happened to fall in the 10-15% that was relapsed refractory. In that relapsed refractory population, about 70% of patients will be, maybe 80% of the patients will be cured with CD19 therapy. Now, what's interesting about it is that for some people, it doesn't work because the cancer cells basically evolve and mutate and chuck away the very recognition system that allows them to, the T cells to recognize them. So they basically chop off the piece that allows the T cells to recognize them. So that's one very common mechanism of escape. And then you can't use CD19 anymore. So that's the understandable piece. But there are some relapses, I just actually saw one, but there's some relapses that are CD19 positive that still have the whole protein in them, but for some reason or the other that we don't understand, are escaping the T cell response. And they're really mysterious ones. I just saw one, actually, bizarrely enough. A patient with CD19 positive cells got CD19 therapy, got a CAR T cell directed against that. We examined, had a relapse, florid relapse. We examined her cells, they still have CD19 on them. So thankfully, she had a second dose. So we always make 2 doses. So she had a second dose. She's getting a retreatment because she qualifies for retreatment. And so we're hoping, that with the second treatment that we will get those CD19 cells. But there's some mystery there that we haven't yet solved.
Do you think we will see human immortality in our lifetime, or is it, as you say, like another seductive idea?
It depends on what you, I suppose it's quite clear what immortality is. I think we'll see some variant of soft immortality. I have an essay that I'm sort of working on, which no one seems to like. But it's a thought experiment, and I thought, let me suggest it to you, just for fun. So my thought experiment is the following. You see, data gathering and data collection has, and data storage has become so inexpensive that we can essentially store infinite amounts of it. So that just let's remove that as a consequence or a variable. Now, imagine that you clone yourself, which lies within the reach of biology. So human cloning there's no fundamental barrier against or that prevents us from cloning humans. There are practical ones, but there's nothing in biology that says you can't make an exact copy of yourself. So imagine that you clone yourself and you download using perhaps very, no fancy tools, so no cybernetic inserts into your brain, but just play a movie of your life let's say every 20 minutes of your life, every 40 minutes of your life, or maybe it's on continuous roll as you move through your life, this clone. So you have a genetically identical human who is on continuous data playing back your life as you lived it. That clone also has his or her own free will, so they might decide to stop the movie at some point of time. But let's say they don't stop the movie, but in fact assimilate the movie somehow. That's a kind of chilling immortality. But I suspect that that some people might want to try it out. I'm not for it. It's a thought experiment.
Let's try it. Why not?
Exactly. I'm not going to try it, but it's a thought experiment. But if you mean in the formal sense, will we see human immortality in our lifetimes? I think the answer is no. Yes, right in front.
Based on all of your experience and also from writing the book, do you think cancer will ever be cured?
So I think some cancers are already being cured. I gave you the example of Emily Whitehead. We're now reaching almost 95, 97% cures for acute lymphoblastic leukemia. I think we'll cure, we're certainly curing many other leukemias and lymphomas. In our lifetime, I think we will see the cures of at least 5 or 6 more cancers. Obviously, we'd like to prevent them. And I think what's interesting is that the amount of work that's gone into curing cancers is disproportionate to the amount of research and work that's gone into preventing cancers. And part of the reason is not just, it's not a machination. It's just that we haven't really found, I would say, profound ways of preventing cancer. We know how cancers arise. Certainly we found ways of preventing viral cancers. Cervical cancer has a vaccine against it. It's caused by a virus called human papillomavirus. There are other cancers that are caused by viruses. We can prevent those either through transmission or others. But I think that and this is a surprising fact to me, certainly, since it's been really about 20, maybe coming up to about 50 years since we found, and I'm going to use my words very carefully here, a major preventable human carcinogen. And that last one was smoking. We found lots of carcinogens, but they aren't major in the sense their impact on human populations isn't huge, or they are not preventable. Aging is a carcinogen of sorts. It's not a preventable one. We'd like to age. And so the last one we found is not even, is a squiggly one, which is obesity. And its connection with cancer is really quite unclear. So what I would love to do is to explore now with new molecular tools that we have and new epidemiological tools and new computational tools that we have to really figure out ways to delay and thereby prevent cancers arising in our lifetime while we concentrate on cures. And I think if we do that, I think we will make a lot of progress. And it's not going to be, eat more goji berries. It's going to be something about some marker that you have, a genetic risk that you have that we can actually, and their sequencing is not seduction, a genetic risk that you carry, probably not in one gene, but maybe a combination of 150 genes, stratify people based on that risk, and assess those people using whatever that risk is, try to find mechanisms to prevent those or detect those particular cancers. Now, I say all this with a little grain of terror. In fact, I would encourage you to read a piece that I wrote it's in the Wall Street Journal, called Welcome to Cancer Land, or something like that. I forget the title. Because it essentially makes everyone a patient. You assess a risk, and since cancer is such a prevalent disease, and a decision we need to make as a society is if that's the kind of society that we really want to be in which you are always being assessed for risk and your life lives in the shadow of risk. And this may be not just for cancer, but for other diseases as well. And I've written about it. I've written about it in a way that I hope provokes some thinking about what it would mean to enter this kind of world of risk. There's a very chilling word that patients with breast cancer or the BRCA1 gene use. They call themselves previvors. A previvor is a survivor of a disease you haven't yet had. It's a very bizarre word. It's a Kafkaesque word. Inventing a world of previvors, not just of cancer, but of all other diseases, would be very distorting for society. It would create a perpetually anxious society. So it is not clear to me how and whether we should dip our toes in that world or not. Now, I know I gave you a very complicated answer to your very, very simple question.
Sid, I want to ask you a total non sequitur question.
Okay.
I believe it was a piece in the New York Times or the New Yorker, one of your fancy New York publications, that followed you in a day of the life. I don't know if you remember this.
Okay.
It's like, so what does Sid and his family do on a Saturday?
Right.
And you did all of these very sophisticated things. You went to the museums, you went to the bookstore, you cooked, and then...
How do sophisticated cooking is sophisticated?
Okay. For me. In the afternoon, you did something shocking, which was you, your wife, and your children read for 2 hours or something like that. I've got a 2-year-old, and I'm curious how you created that discipline.
That monster.
Yeah, I mean, truly, how do you get kids to read?
What's interesting
how does the whole
is
family read every day?
we've brought up, I have to say, we've brought up 2 readers. And they are real readers. I come back, it's kind of amazing. They are real, I have 2 daughters, and they are real readers. I mean, she'll come to my older daughter in particular, who's 17, will come to the dinner table and I'll say, I just was reading this piece or I'll reading this thing by, I don't know, someone, Jenny Egan or Zadie Smith or someone, and she'll say, oh, I've read that. And I'll say, oh, what do you think about it? And she'll go on to tell me what she thinks about it. And she's prolific and she reads everything. Now the question is how or why. I think it's because I think you make a reader never by telling anyone to read anything. I've never seen anyone become a reader by being told to be a reader. It's like cats. The only way they became readers is because we were readers.
Wait, how is it like cats?
It's like cats because when you tell them to do something, they do the opposite.
Oh, okay, got it. Okay.
But I think the way you bring up a reader is by reading. And that's the only way you can do it.
Was there a moment where you were reading and they weren't reading? And
I don't remember. I mean, I think, actually what's funny about it is that we didn't even particularly read to them.
Huh.
We just read our own books.
Right.
Selfishly. It wasn't as if, we said, oh, look how fun it is to read, and then they started reading. And no, we just said, it's an afternoon, how nice it would be to read my own book, and you go and do your own thing. And eventually, I think they just got tired of roadblocks, and
yeah.
and said, actually, the world of imagination that reading brings is more interesting than putting blocks on top of blocks. And they started reading. I can't tell you the secret. All I can tell you is that I've brought up 2 incredible readers.
Next
Sorry.
question. Maybe we have, I don't know, Ted, how many?
So, I'm not sure how to ask this, but science has taken a huge hit in the last 4 or 5 years
Well, that's a good way to ask the question.
for very many reasons. And I'm just wondering, how do you keep the faith in mankind, like you keep your faith in cellular science or biology or, whatever, knowing that we could just as easily end ourselves, on big scale, little scale. How do you keep that positive, faith going when somebody says to you, "Oh, I don't believe in science"?
Well, I think that the answer lies in the fact that, generally speaking, people who don't believe in science, when they encounter adversity, such as the adversities that are coming with climate change, or the adversities that come with personal medical issues, the adversities that come with changes in lifestyle and behavior, they reach a kind of mental compromise in which they begin to understand that there is value in medicine. I'm a doctor, so it's going to be a doctorly answer. I think that people make a compromise, and they say, "I'll believe a little bit of science," when they face these adversities, and they're coming all the time.
People were in the hospital with COVID.
Yeah.
Denied they had COVID.
Yes. Yeah. I mean, look, you're not gonna. You're certainly not gonna get the answer from me that I'm totally sanguine about what's happening in the United States and the world right now. I'm not sanguine about it at all. But I do think that medicine is a good thing. I think that the intense bravery of physicians, and more than physicians, really nurses, was grotesquely under-recognized during the COVID pandemic, and for political reasons. I would have made last year National Nurses Year of the Nurse. I would have, there was something very moving every evening in New York at 6 o'clock. People would open their windows and bang their pots to applaud the nurses and doctors who were sitting in the front lines with full knowledge that they would get COVID and might die from COVID. There was something very moving about that. I'm saying this not because, as I said, I'm not sanguine about the world, but I'm also saying this because I think that I suspect that some of these will stay. People who are deniers are deniers, but I think some people will convert over their lifetimes as they face personal adversities that come with that come naturally, and they'll reach a kind of soft compromise. But that said, the mental and physical toll that COVID has taken, particularly on the medical community, the burnout that we've seen among doctors, really needs to be addressed, and it's making it a political issue, and denying it is just really grotesque. I'm sorry to say.
And our final question for the evening.
Back on the cell, you mentioned we thought it was rare that a single cell could be multi-cell, but it turns out that it's happened more than once and many times throughout history. It's proven to be, it seems, incredibly rare that we had an original cell, a single cell, kind of come out of who knows where. And we're looking on other planets, maybe that happened. Can we be looking on our own planet? Is it possible that we will be able to create like a separate track? You said we've never made life except from former life, but is anyone working on this? Is it an interesting problem to solve? Do we even know where to look? How did it happen in the first instance?
Yeah, so lots of people are working on it. I give some examples in the book of people working on it, which are fascinating. People are working on duplicator molecules, self-duplicating molecules. People are working on self-duplicating or dividing membranes. So all of that absolutely is being worked on. The problem is that I think that it depends on what the purpose is. If the purpose is to satisfy natural curiosity, I think it's perfectly reasonable to try to make a new kind of cell from scratch. So life not out of life, so-called abiogenesis in the fancy words of biology. But if the purpose is to manipulate life to make it do things that it hasn't done before or it can't do before, which is a much more engineering-like purpose. So for instance, to eat up leaked oil in the ocean, then you're probably better off starting with life and re-engineering life because it's a much easier thing to do. So it really depends on what the purpose is, what question you want to ask. Because it seems to me that the amount of manipulation that you can do with life itself, simple forms of life, starting with those simple forms of life, injecting, for instance, a totally new genetic code into a simple form of life, is very powerful. The synthetic biology universe is very powerful these days. So I think there's a lot there. But if you want to answer the question, can you create life from no life? It's a fascinating question and may have many future uses. But it seems to be right now more of a kind of scientific curiosity rather than a purpose-driven experiment. There's nothing wrong with scientific curiosity-driven experiments. Lots of people do them. I give examples of people who do them, but it's a different kind of question. And so I think lots of people are trying to make self-duplicating molecules, to make splitting membranes, to make those self-duplicating molecules carry information that would potentially create metabolism of a very basic sort, using basic nutrients to make more complex molecules. So I think all those questions are absolutely fascinating. But on the other hand, there's also so much to do with engineering life that already exists and using new genetic material and inserting new genetic material into it, changing the genetic material completely, that is also equally interesting and perhaps more applicable in the short term. Well, I think
Thanks, Sid. It was a pleasure.
My pleasure.
Thank you all for coming.
Thank you very much.